Good afternoon, everyone. I do believe that I can start my presentation. I will ask Diogo if he could share the screen. Okay, so good afternoon again, everyone. My name is Carla Torre. I'm from the University of Lisbon in Portugal, and we are one of the partners of this beautiful consortium, More-EUROPA. And I will explain a little bit for those who don't know already, what is our consortium and what are our main aims, I will explain later. So this is our third webinar, which is entitled How Can Real-World Registry Data to be Used to Augment Clinical Trial Data to Improve Drug Development and Regulatory Decision-Making. Next slide, please. So some housekeeping rules. So all participants will be muted. The chat box is disabled, but please, if you want, and we really enforce you, because we would like to be this a kind of enriched session with your questions, with your comments, with your suggestions. So please add your questions in the Q&A box, and they will be answered or during the plenary, during the webinar, or at the Q&A session at the end. So for transparency, this webinar is being recorded, and we will share the recording with all participants after the webinar. And this was also the case for the first and for the second webinar. So all the webinars are placed on our website. Next slide, please. So in terms of the agenda for the webinar today. So I'll give a flavor, I would say, about the project, but the most important are our distinguished speakers. So the first speaker will be Kelly Pluche from the European Medicines Agency. Kelly will share insights how real-world evidence can contribute to marketing authorization applications. So a landscape over the last few years with concrete examples, and how this real-world data contributed somehow for the marketing authorization regulatory decision-making, I would say. Then we will have François Houy from the European Organization for Rare Diseases, EURORDIS. And François will present a patient perspective on the real-world data. Then we will have Kit Rose. Kit Rose is a professor of biostatistics at Red Bull University Medical Center, and he is also chair of the Methodology Working Party at European Medicines Agency. And Kit will discuss the role of registry data in early development decision-making. Then we have Q&A session, and again, I strongly suggest, I reinforce again, so to place your questions in the box, in the chat box, for this dialogue could be somehow more rich. And finally, this Q&A session will be moderated by Kit, who will unfortunately, Peter will not be able to be present, but Kit will wrap up the session and state some conclusion statements. Next slide, please. Next. So More-EUROPA, for those who are not familiar, this is an European Commission-funded project. This project aimed to establish the value of registry-based real-world data in augmented RCTs. And also, and that's where the name comes from, enable more effective and ethical use of registry data to support what? Patient-centered regulatory and health technology assessment decision-making. So has this kind of dual approach in terms of the regulatory action and in terms of the HTA action and decision-making as well. Next slide, please. So to achieve this, so the project that we are developing, somehow entails a comprehensive methodological framework, which somehow includes analytical and screening tools for registry data assessment, and that. As you can see in this slide, we have a kind of a complete landscape of the medicines development and in terms of the decision making as well. So our main aims is how to provide some tools and how to better enable the use of this data to a better contribution to the decision-making process. And as I mentioned before, both at regulatory or HTA payers level. Next slide, please. So we have this partner is this consortium, is composed by 15 organizations across Europe, public, academic, private organizations. So I think patient representatives. I think the best flavor of our project is somehow because we are different nature in terms of organization and how these different organizations across Europe can somehow contribute to this active discussion and propose solutions to achieve our main aims. Next slide, please. So in terms of the packages, so this project is structured in 5 working package, and I have to read my notes because I don't know the numbers and the names by heart. So the first one is to develop the working package 1, is to develop the novel analytic tools. The second working package, the working package 2, establish real-world data accessibility and usefulness. The working package 3 is to identify and screening suitable registry data. The working package 4 is to explore ethical perspectives and patient empowerment in registry data use. And the final one will be the dissemination one. So how to disseminate these deliverables, these outcomes in order to boost and to contribute for this use of registry data for regulatory and HTA payers decision making. And one of these main outcomes, I would say, with this working package 5 is also to develop a kind of a cascade of different webinars and to dialogue with the community in order also to share our deliverables and to get input from other scientific and regulatory and HTA community. Next slide, please. So this, I will not go into detail for this publication. So the next 2 slides are different deliverables that are presented also in our website, and I will leave the link in our last slide, but just to give you somehow a flavor of our project and some of these publication deliverables. And you have on artificial intelligence, and we have concrete use cases through the multiple sclerosis registry, from the heart failure registry, etc. So we have these 3 concrete registries example, and we try to build also on this registry data. Next slide, please. And my last slides that I would like to share with you. So at our baseline, I would say, we conducted a survey among regulators, among HTA payers, among academics, industry, etc., in order to collect some data about, for instance, and I have these 2 questions here, because we thought that could be important to share with you these first outputs, and we have a paper that will be published very, very soon about this. And we ask the community how informative, so at baseline, because we intend to conduct several surveys, so at baseline, how informative are registry data? And we put some options or several options in terms of disease epidemiology, long-term effects, etc. So here you can see in the slide. So while and our main conclusion was, while stakeholders seem generally open to increase the use of registry data, they somehow perceived some weaknesses and strengths. In terms of the strengths, they highlighted, or in terms of the use, the disease epidemiology as one of the most important use, whereas, for instance, the comparative effectiveness was one of the domains with where they couldn't see at least this value in terms of the use of registry data for the decision-making process. Next slide. And in terms of the strengths and the weaknesses, how I mentioned before, all these stakeholders seem to perceive the data quality as a weakness in order to use these registry-based studies, information to support the decision-making, but they thought it could be useful in terms of capturing data and capturing outcomes, for instance, for different populations who are not, for instance, included in the clinical trials. Next slide, please. So our previous webinars were about AI, artificial intelligence, and machine learning approach to identify and appraise registries and relevant data. We had one use of registry in the regulatory decision making, and here in this slide you can have the links and you can access these recordings, because for us it's part of this dissemination working package to, as the word mentioned, to disseminate the knowledge. So the knowledge should not be in the box, should be disseminated within the community. Next slide, please, and this will be my last slide. So here we have, for those who are not familiar or do not know our consortium, so here you have the link where you can access, when you can see some of the deliverables so far, and could be, and you will be updated on the further deliverables about our project. So now I hand on to Callie, and thank you very much for being with us.
Thank you very much, Carla. Good afternoon, everyone. I'm going to share my screen. Yep, share. Oops. Can you see in presenter mode?
Yeah, maybe Kelly, also just before to proceeding, just to mention to the attendees that there is possibility for clarification question after each presentation. Hence, please feel free to ask the question in the Q&A box. Thank you.
Thanks, Cécile.
You can see the screen, yeah.
Sorry?
You can see the screen.
Okay, perfect. So good afternoon, everyone. I'm very happy to be here with you today, together with my colleague Anna Razokat from the University Clinic Köln in Germany, to give you a little presentation on a review that we've performed, which looked at real-world evidence included in a medicine dossier submitted to the European Medicines Agency. So this is what we're going to talk about today. So we give you a little bit of background to explain about the reasons for these studies. Then we look at the contribution of real-world evidence in the submissions to the agency and their contribution to decision-making. First, for the period of the application submitted between 2018 and 2019. And then I will hand over the floor to Anna, who is going to give you some insight into the latest period follow-up study covering 2020-2023, so preliminary results. And then we'll finish with some key messages for the future. So as you may all know, there are several regulatory science strategies which have been developed and published on the EMA website. There is, first of all, the strategy of the EU regulatory network and the one of the EMA. The ones you can see on the screens are the ones that were published before, so in 2020, I think, for the strategy towards 2025, because these were the ones available at the time we were planning for these studies. Obviously, nowadays, there are strategies which have been updated and published. I think if it's not done, it will be done very soon, strategies to 2028. But these are the ones we were considering at the time. And these strategies are aligned, obviously, so, and they both highlight the need to promote use of high quality real-world data in decision making. They also acknowledge that real-world data have been predominantly used in post-authorization phase, but they really represent opportunities. They really could be useful, to complement clinical trials, and we should really embrace and leverage these data to address limitations of clinical trial and support decision making. So in order to achieve these objectives, we thought that it would be very useful for us to better understand first what kind of real-world evidence is submitted to the agency, and at which occasion, and with what level of quality, and how does it actually contribute to decision making. So that's why we started with two first studies, which you can see here on the screen, looking at the contribution of real-world evidence included in initial marketing authorization applications, as well as extension of the indication applications submitted to the agency, to the EMA, in 2018 and 2019. So the first study was published in October 21, and the aim was to characterize real-world data and real-world evidence submitted to the agency, again, for the 2 years period in initial marketing authorization and extension of indications. You can see here the exclusion criteria. And then the second study, a follow-up study, was performed and published 1 year later, where we looked at a subset of the products from the first study, whereby for which the real-world evidence was submitted to support efficacy. And for these products, we really drilled down, we really went in depth into the assessment of our scientific committee, the Committee for Human Product for Human Use, the CHMP, to see how the CHMP actually appraised this data and how it used it for assessment and decision-making on medicine. And the CHMP, just to note that this is the committee which provides opinion on whether or not the product should be authorized. The third exploratory objective was also to identify any gaps in guidance on the use and evaluation of real-world data, real-world evidence. So these were the working definitions that we used at the time of the study. So back in 2020, 2021, we considered real-world data as routinely collected data relating to patient health status or the delivery of healthcare from a variety of sources other than traditional clinical trials. And these included, of course, data from patient registries. Real-world evidence was considered as the information derived from the analysis of real-world data. Since then, these definitions have slightly have been updated, and you can see the new definitions in recently published guidance document. But this is what we used at the time of the study. In terms of inclusion and exclusion criteria, we considered applications including real-world data, those which included non-interventional studies conducted either pre or post-authorization, which were based on real-world data used, for example, to contextualize the results of clinical trials, real-world data providing insight into the disease epidemiology, like incidence, prevalence, or also real-world data that showed how the standard of care were used, which provided insight into the patient characteristics, for example, or also data that provided support, that provided information on the effect, so including efficacy and safety of the product. We also included in this in this study the applications which included real-world data as external control arm for clinical trials, and the surveys addressed to patients. But we did not consider applications as including real-world data if their clinical trials did not include real-world data, obviously, or open-label follow-up studies of clinical trials, as well routine pharmacovigilance activities in risk management plan, and also the surveys addressed to healthcare professionals. So in terms of the methods, what we did is that from the document management system at EMA, we identified all the submissions that we received for initial marketing authorization and extension of indication, which were submitted to us in 2018 and 2019. So we extracted all these products into an Excel table, and for all of them, we retrieved various documents, including final versions of our scientific committee assessment reports, CHMP assessment reports, the risk management plans, and also the protocols of studies based on real-world data. This helped us to characterize the real-world data and real-world evidence included in this dossier. We used an online-based form to extract the information and to structure information for analysis. And we also had 2 independent reviewers to ensure data quality. So in terms of study for the first, in terms of results, sorry, for the first study, as you can see on the screen here, we identified 158 applications for a new marketing authorization, so initial, out of which 63 of those applications included some form of real-world evidence. Most of this evidence was proposed to be submitted post-authorization. So 40%. And for extension of indication, we identified 153 products, out of which 28, so 18%, had real-world evidence submitted, either as part of the dossier, so in the pre-authorization phase, or proposed to be used to be collected post-authorization. And quite a lot of these products, which included real-world evidence, covered indications for oncology type of diseases. So what about registry data? As you can see here in blue, out of the 63 and 28 applications which included real-world data, respectively for MAAs and extension of indication, for MAAs, more than the half actually included registry data, and nearly the half for extension of indication also included some registry data. But mostly this data was proposed to be collected post-approval. Now, a few words on the second study. So in terms of method, we used the same, so we used the data set from the study 1, as you can see on the bottom left on the slide. So I'm back to the 63 and 28 products, which included real-world evidence in MAAs and extension of indication. And what we did is that we drilled down within this application to find the ones for which real-world evidence was submitted to support efficacy, and also more specifically efficacy in the pre-authorization phase. And so we found 16 applications for initial marketing authorization applications and 10 extension of indication applications, which included real-world evidence for efficacy in pre-authorization phase. And for these 26 products, sorry, we went back to the CHMP assessment report and really went more into the details in depth of the appraisal from the CHMP in order to identify what were the strengths and the limitations for the real-world evidence submitted and how this actually contributed to their decision making. We also had some quality check by 2 other reviewers. And at the end of the analysis, we identified 5 applications for MAAs and 5 in extension of indication, which were considered, for which real-world evidence was considered really contributing to decision making. So at the end, it's not many products, but still real-world evidence can support the evaluation of benefit risk of medicine. And on the right-hand side, we've put here the number of products or applications for which registry data were submitted to support efficacy in the pre-authorization phase. So we had 5 applications for MAAs and 3 for extension of indication. So not many products. So covering this period 2018 and 2019, we did not identify any initial marketing authorization which were submitted by which supported decision making. So no registry data supported decision making on new marketing authorization. However, for free extension of indications, registry data actually contributed to the decision making, so supported CHMP evaluation. And for two of the products, they are for rare hematological conditions, heptacog alpha and Cadre-CACOG. Registry data were submitted to support efficacy and safety effects or safety claims, as well as they provided insight into the natural history of a disease and also the use of a standard of care. So this helped contextualize the results of single-arm trials. And for ivacaftor in cystic fibrosis, actually registry data was used as external comparator, and it was considered as well to be useful for decision making. And just a note that within the CHMP assessment report, what we have identified is that even if the registry data, real-world data, were considered supporting decision-making, mostly in the assessment report, the limitations are more explained than the strength. So they are more acknowledged than the strength. For example, limitations linked to the missing data, the lack of representativeness, the different types of biases, etc. So these are much more explained than the strengths of the real-world data. So I think I will now hand over to my colleague Anna to give you some insights or preliminary results of the follow-up study, which covers the period 2020 to 2023. Anna, over to you.
Yes, thanks very much. So I will present the follow-up study covering the period from 2020 to 23. We used the same study design as for the previous study, also the same inclusion and exclusion criteria. But we slightly adapted the process, starting with an automated search for the key terms on the left-hand side of the slide, then followed with a quality check and a manual data extraction. And this last bit is still ongoing. The final data set comprised, one slide back, please.
Sorry.
I'm sorry. The final data set comprised 244 applications, 318 were initial marketing authorizations and 426 extensions of indication. And for 42 products, there was no assessment report available yet. So the evaluation process is still ongoing, and we are going to update the data set like in the coming weeks. But that, of course, means that the results that we are now presenting are only preliminary. Next slide, please. From what we've analyzed so far, we can see a clear increase in the use of real-world evidence compared to the previous study period. In initial marketing authorizations, 58% of applications used real-world evidence, mostly though in the post-authorization setting. And for extension of indications, it was 56% compared to 18% in the previous study. The majority of products with real-world evidence in the pre-authorization context pertain to the same therapeutic indication as in the previous study, so kind of cancer-related, antineoplastic, and immunosuppressants. Next slide, please. I would like to present a few examples where real-world evidence was included in the application, just in order to highlight a few strengths and limitations for this type of evidence. So in the first example, we have an extension of indication for a drug used to treat metastatic colorectal cancer for those patients where the standard of care treatment is not available due to intolerability. And the purpose for the use of real-world evidence was efficacy or effectiveness. and the submitted evidence combined a meta-analysis with 2 observational cohort studies. One of them was linked to the Dutch National Cancer Registry. And in the appraisal, it was acknowledged that an RCT would not have been possible in this context where the lack of, or the fact that the standard of care treatment was not available for these patients meant that there was no possibility of a proper control group. RWE was considered supportive in this case, and there are still listed a lot of limitations to this kind of data regarding kind of retrospective and uncontrolled nature of the data, limited sample size, and heterogeneity of tumor types and treatments received by the study populations. And these are criticisms or kind of limitations that we find in many assessment reports with regard to real-world evidence. Then the next example is a treatment of progressive familial intrahepatic cholestasis, where data from a registry was used as an external control. And here it becomes clear that a detailed description of the registry is of great importance. So CHMP asked for further information regarding the registry and its data collection methods. And they then also highlighted fundamental limitations of indirect comparisons, such as residual bias, which cannot be quantified. And therefore, real-world evidence was not deemed appropriate as pivotal evidence for efficacy, but it was considered supportive. Then the next example is a treatment of unresectable or metastatic HER2-positive breast cancer or gastric cancer. Here again, real-world evidence was used as an external control arm with historical controls from the matched uni-cancer cohort, which is a cohort that contains routine clinical data from French hospitals. Again, here the concerns with real-world evidence that are listed in the assessment reports can be found in many, with regard to many studies or applications. So the matching was considered inadequate regarding the comparison of baseline characteristics in the trial population regarded in comparison to the external control arm. And then there was a lack of information on timing and follow-up scans regarding the assessment of tumor progression or tumor status. On the next slide, we've got 2 examples where real-world evidence was used successfully. So in the first example, real-world evidence is used as complementary to a clinical trial. Here we have a pivotal study where CHMP criticized its limitations due to a small and uncontrolled sample size. But the real-world evidence that was submitted actually complemented the data so that the general efficacy statement was supportive. And also with regard to exposure time, it was mentioned that the pivotal trial had only considered an exposure time of 6 months, whereas the real-world evidence provided a follow-up time of 8 years, and therefore could reveal a common cause of drug discontinuation, which was not apparent from the trial data. The final example shows the use of registry data to contribute to the safety evaluation in the case of the monitoring for birth defects for a drug of pre-exposure prophylaxis for HIV infection. And here data was used from an antiretroviral pregnancy registry, and that registry will also be continued post-authorization, and it should be highlighted that this registry constituted a collaboration of several companies rather than being a single product registry. Next slide, please. So here I would like to show you a few examples which demonstrate the growing awareness of the value of real-world evidence that we could see in the assessment reports that we looked at. So there is one example in filgrastim where the company actually ended up not including real-world evidence in the final application, even though that had been discussed in the previous kind of scientific advice sessions with CHMP. And in the assessment report, it is then criticized that this real-world evidence was not supplied because it might have given important information on the contextualization and might have shown an unmet clinical or medical need, which was not supported with the data that was actually submitted. So real-world evidence might have been required in this case. And then another example, Brianzi, where EMA actually encourages building an infrastructure for real-world evidence collection with regard to CAR T-cell products. So it is deemed important to have a platform to collect RWD regarding a technology-specific, with regard to a technology-specific registry for regulatory purposes. So yeah, thank you very much. And I'm handing over to Kelly for some conclusions.
Thank you, Anna. I think it's useful to have these kind of concrete examples that speaks a bit more, at least to me. So some key messages out of these 3 studies. We've seen that real-world evidence, so including also from patient registries, can contribute to medicine's benefit risk decision making. There are so far this type of evidence is considered a lot supportive, but still I think we can really identify some opportunities here, and we can work a bit more all together to try to really extract all the potential of this data, also to be used in the future more for efficacy pre-authorization. Real-world evidence is actually part of the overall evidence package, so it's not always easy to isolate its impact to decision making, and it also depends on other factors. It's influenced by other factors. For example, results of pivotal studies, if they show really high level of efficacy, real-world evidence might be, more easily accepted in a way than the contrary. The appraisal of real-world evidence requires really a case-by-case analysis to really appraise and see whether it's fit for purpose in the specific context of use. And for this, we've highlighted here on the screen 2 really important aspects. The first one is really the need to perform thorough feasibility assessment prior to writing a protocol and starting a study, in order to really assess and understand the opportunities and the limitations of the real-world data, to see if it's really relevant to provide the necessary evidence to answer a specific research question. And here we've put some examples of guidance which are available to help perform or conduct this feasibility assessment, and there are possibly others, obviously. And the importance of early interactions with regulators. This can be done at a very early stage of design of the study, and this will help really understand the data needs, the evidence needs, but also understand the challenges that can be encountered in assessing the feasibility of using real-world data, and also trying to work together to see how these limitations or challenges can be addressed. And we put here the link to various interaction pathways, which I will show a bit more in details in the next slide. There are more guidance documents that will be developed in order to enable the use and feasibility, facilitate the integration of real-world evidence in regulatory decision-making. So we put here the link to the HMA, so Heads of Medicines Agency, EMA Big Data Initiative, where you can see a lot of activities ongoing there, as well as the work plan of our methodology working party, which includes, I think, insight into the guidelines that are planned to be developed over the next 3 years. EMA interaction pathways, so stakeholders can reach out to us and the regulatory network in order to get support on regulatory and scientific aspects for various topics, and this is to foster development of new and innovative medicines at a very early stage of development. We've put here the links to various routes, including the EU Innovation Task Force. There is an academia office as well, an SME office, small and medium enterprises, the PRIME scheme for medicines with potential to address really high unmet medical needs. Also the qualification route, whereby applicants can get an advice or an opinion on the novel methodologies, and this can be also applied by patient registries in order to create a dialogue with regulators on their registry data and to see how this can be used for regulatory purposes. And finally, the scientific advice and protocol assistance route, which is also available on study designs, for example. And important to know as well is that there can be a fee waiver for protocol assistance for academic organizations developing orphan medicines. And this is our last slide. We thought we'd just highlight here a bit more details on the EMA qualification of patient registries, which, like I said, it's a route that can be sought by patient registries in order to create a dialogue with regulators in order to see how their data could be used or support regulatory use. So far, there are 8 registries/data sources which have come for qualification, and you will have the links on the screen for the qualification opinion and advice. And what we wanted to say is that it's very important to note that this qualification is based on a specific context of use. So meaning that registries can come speak to us on their on how to use their data, for example, in the context of post-authorization studies, or in order to provide useful information on drug utilization patterns, etc. We do not qualify patient registries as a whole, as an infrastructure. We qualify them based on the context of use, and there are information to be provided in order to facilitate the assessment. This qualification does not replace the feasibility assessment step, which I highlighted in the previous slide, which is linked to a specific research question. So here we would qualify for a specific umbrella use based on the context, specific context. Qualification is voluntary basis, so obviously it doesn't mean that if a registry is not qualified, it is not good for purpose to be used for regulatory purposes. But it's a good way to come speak to us and assess the registry data. There is currently a review of the qualification guidance for novel methodology, including for patient registries, in order to optimize the process and to make it clearer in terms of scope, added value, benefits, and outputs as well for qualification of these methods. And we've put here the link to the report of our last workshop on patient registries, which include much more information on this topic. Thank you very much. I think we will stop here. And thank you, Anna, as well. And happy to take any questions if you have any. Thank you.
Thank you, Kelly. Actually, there are a few questions in the Q&A, and maybe one is interesting that is coming back regularly is the connection with Darwin and Sami Pakarinen is asking, had these real-world evidence sub-studies been conducted with harmonized data and federated analytics within the Darwin network?
No. So Darwin is something totally different. So we have identified here all the real-world evidence or real-world data / real-world evidence submitted by sponsors, so by marketing authorization applicants. Darwin is a federated network of data sources, which is managed by the European Medicines Agency, and it is not accessible by industry, pharmaceutical industry at the moment. And we use Darwin to support our EU regulatory network, to support our scientific committees in their decision making. So these are totally two different type of, application of real-world data. We focused here on our 3 studies based on the, the data submitted by industry. And I really forgot to say as well, just to acknowledge our colleague Lisbeth Baker, who has actually been running, or she has done the analysis of a study to the second study, and I think she's on the line now. So thank you very much.
Thank you. And then there is a question from Diogo Lopez, who is wondering if there are data orphan versus other therapies in the studies.
Yeah, so in the first study, we have identified, we have characterized the type of products for which real-world evidence was submitted. So we identified whether the products are orphan, whether they are ATMP products, whether they had the prime status, and all the type of therapeutic indications for which the application was submitted. And I think you will find more details in the papers published.
Thank you. And there is another question, and maybe it can be answered in writing, if not briefly here. In what format does the EMA wish to see the results of a feasibility assessment, whether it's a report, document, slide deck, a spreadsheet, or as part of a protocol? Do you have any guidance on that?
That's a very good question, and maybe some colleagues on the line are better placed to answer. But this is definitely not as a slide deck. I think it needs to be properly structured, and we've put some links to guidance on the slides. There are some templates that exist in order to, for the studies, for non-interventional studies based on real-world data. So this can be, a good start. In the different guidelines we've highlighted as well, there are some, structured information that should be submitted. So I think colleagues or colleagues on the line can really refer to this guidance, and they will find there are some ways to structure the feasibility assessment. And I think this should be submitted to the agency as a proper report, in line with regulatory procedures that they support.
Yeah. Thank you, Kelly.
Thank you.
There is another question, but I think we need to move to the next speaker, and Thomas Klopstock will answer the question in the discussion. That's an interesting one, and I'll give the floor back to Carla. Thank you.
Thank you.
Hello, everyone. I think that we can move for the next speaker, Francois. Thank you very much for being with us.
Thank
Thank you, Kelly, for your brilliant presentation.
Thank you very much, Carla, and hi to everyone. So I was asked to address the perspectives of patients for real-world data and their interest for registries. And as this is a very broad aspect, I would like to focus on how patients position themselves vis-à-vis clinical trials or vis-à-vis registries or other sources of real-world data. So I work for ORDIS, which is the European Organization for Rare Diseases. some disclosures. I am also a member of the Get-Real Institute, the ACT-EU multi-stakeholder platform, and the DCT implementing working group. Some introductory remarks to start with. And this is some data that we've been commenting for quite many decades already. I haven't seen more recent data, and this was debated at a meeting of the Pan-European Regulatory Forum back in 2004, I think, when the EU was preparing the entry of accessing member states. And we had quite a lot of projects and programs to prepare this, and we discussed the clinical trials and the participation of patients in clinical trials. And one of the information was that in 40% of cases, when patients refuse to join a clinical trial, and this is because they don't find the design of the clinical trial attractive enough. Maybe this has changed. I haven't seen more recent data. And yet they wish to take part in research activities. There are too few patients involved in research activities, and yet many would like to. So are registries or other cohorts of patients databases an alternative to clinical trials? Probably. There is also some confusion in the mind of many patients between the access to a clinical trial and the access to an innovative treatment. As an illustration, this is not CTIS, it's another initiative to help patients find the clinical trials of interest to them. And although it is a tool to find clinical trials, see how it is called, find a treatment. Clinical trials, accessing to a clinical trial is not synonym of accessing a new treatment. And this is still confusing in the mind of many. When joining a clinical trial, most patients hope to benefit personally. The majority is also interested by helping others. They experience pride regarding this contribution. And we have this debate. There are quite many papers published on the fact that the survival is improved just being on a clinical trial as compared to not being in a clinical trial. Is it because you are better taken care of in a clinical trial? Is this an illustration of recruitment bias? It is not clear, but many patients have this in mind, that joining a clinical trial may help improve survival. In rare diseases, the chances of joining a clinical trial are not that high. And based on a recent analysis by the FDA on the numbers of rare diseases that have seen at least one drug quite advanced in its development, reaching phase 3, this was the case for 11 to 15% of the rare diseases in 40 years of the Orphan Drug Act in the US, which means that 27 rare diseases see a product in development for the first time each year. So quite a restricted offer for research projects for the patients interested to join. And joining a clinical trial is a difficult decision to make, and there are even more difficult decisions that can be made. Here, just 3 examples. In amyotrophic lateral sclerosis, as you might know, the life expectancy is very short, 2 to 3 years, median life expectancy. So when you are proposed a clinical trial with 50% risk of receiving a placebo, and the clinical trial duration is of 2 years, then you may be reluctant to join that trial. So that is why the patient organizations and learned societies are working on platform trials to reduce the risk of being in a control arm, in the control arm. But it makes it even more difficult for those randomized to the control arm, as they thought they had more chances to be in an active treatment arm. In Wiskott-Aldrich syndrome, the first 10 patients who received a gene therapy, and among them, those who were positive to SARS-CoV-2 indicated that the gene therapy was successful in regenerating an effective immune system. They had the virus, and yet they were asymptomatic to COVID-19. And for this disease, the alternative option is bone marrow transplant with quite a high mortality rate associated to the procedure. So then discussing a confirmatory clinical trial, would you, if you were a parent of a child with this disease, would you accept easily a gene therapy versus bone marrow transplant? And when patients and families, because in these networks, all parents and families share the information, the a priori knowledge has an impact on the decision of joining a clinical trial or not. There is no longer equipoise in many situations based on extremely effective or apparently effective results from the initial research, which makes it ethically challenging to propose clinical trials to confirm efficacy. In Duchenne muscular dystrophy, the children who are more than 12 years would often reject the randomization to a control arm and would prefer an open label trial or a single arm trial, or joining a registry where they can receive a new treatment and being compared to untreated controls. So these are many situations that we see every day in rare diseases. They are not specific to rare disease, but they are very frequent. So when there isn't a trial, and this is another testimony of a mother who lost her 2 children from a neuronal ceroid lipofuscinosis in 2003. And she reported that losing her child was devastating, as we can imagine. They had suffered for 15 years with their 2 children born at different years, but for the parents, it took 15 years taking care of them 24 hours a day, 7 days a week. And after both of their children passed away, they realized that they were too old to envisage a new life with their children again. And then they learned that no data had been recorded, no tissues had been stored, no registry, nothing, no research could be done to benefit children in the future. So they had the impression that their children had died for nothing. So the setting up and the creation of registries is really the basis to initiate research. Another example where a patient joined their forces with clinicians to collect data and organize this data is Behçet syndrome. And in this disease, for 20 years, they had seen no clinical research, no drug development for 20 years. And then 4 at a time came into discussion, which you can see here. So the European groups, they had in total 300 patients in their membership, and they thought that was too few for the development for these 3 products. They created an online community in 2011, and in less than 3 months, they could identify 1,350 patients totally in the world, 900 of which indicated their location and their interest for joining a registry to organize the recruitment for all these clinical trials. And they went one step further. They asked patients how they treat themselves. And these are some results of a survey that they made within this community in a subset of patients who responded. And the numbers are not high, but you see the different products that the patients took either daily or as needed. So it's a highly medicated patient population, which products which exposed to safety concerns all were off-label, and no data were collected specific data collection was organized on safety and efficacy. Of course, the clinicians could also submit spontaneous reports in case of adverse drug reactions, but there was no registry to collect the data and to compare the efficacy. So they decided to support the creation of nationals and international registries for BESET, on which to build first the comparison of the outcomes with these different treatments, but also to support the conduct of clinical trials. This situation, when there are several clinical trials competing with each other to recruit patients, raises questions. Would we opt for a global registry? Should each company have its own registry? And again, another illustration for Sanfilippo type A disease, where the parents made an audit in October 2005 when no product was in research and development for that disease. And then some companies initiated some research, probably because the disease had reached kind of a maturity, a critical mass in terms of research activities and publications. And then you see the date of designation of several, orphan drug designation of several products by different companies. And then the patient community was facing this difficulty, maybe more products in development, many gene therapies, than there were patients to join the research and development for so many products all at once. And so this really makes the need to have global registries and to use the registries or real-world data to better characterize and identify where are the patients who can join different clinical trials in development in parallel as something absolutely useful. And that also supports the need for early screening, even when there isn't a treatment available. If you are not screened, you may not be diagnosed with a disease, and this reduces your chances of joining research, of joining a registry on which future developments are based. A lot of patients are interested by natural history research, natural disease history research, as the first use of real-world data and registry data. Here, just give you an example for Batten disease, where in Hamburg, Dr. Angela Schulz, with 58 children, could build this composite endpoint, motor and language score, in a devastating disease in children who lose all their cognitive functions very rapidly. And here you can see the slope of decline of this composite endpoint of cognitive functions at the age of 72 months. It's almost a score 0, and that illustrates the devastating effect of the disease. And this endpoint was used to compare with the results of a phase 1 trial with an enzyme therapy in 24 children. And they use exactly the same endpoint which had been used by Angela Schulz to demonstrate the natural disease evolution recently, shortly before this phase 1/2 clinical trial. And so they thought, when they saw the first results showing that very early in the course of this trial, 6 of 9 first patients treated were completely stabilized, which had never been seen before, and which was later confirmed by the other patients. Then the question was, are these data enough? Can we use these phase 1/2 results compared to this very recent and very well done natural disease history to convince to submit a marketing authorization application? And so this is what they did. They had pre-submissions meeting and a submission to the EMA. The question was, is this data enough to support a marketing authorization, or should a confirmatory trial be organized that would support more the marketing authorization? So this is what was achieved. There was no need for a confirmatory trial. The data was so convincing. The comparison of the phase 1 with the natural disease history was convincing to grant a conditional marketing authorization. And this raises the value of information question, which is not often debated, but at some point the decision is, do we have enough information to decide, or do we need to ask for more information, knowing that asking for more information would mean more resources, more investments, more time and patience involved in this research who cannot participate to other research. These are questions that should be systematically discussed. Can we decide and act with a conditional marketing authorization based on this type of development, comparing an open label trial to registry data and natural disease history, and then confirm the effect based on real-world data registries or electronic health records, or the alternative being to defer the marketing authorization and first request confirmatory data. I think the discussion, the debate is permanent, is frequent for many drug development. So what do patients like about registries? As just said, the fact that with them, we can learn more on the natural disease evolution, which is lacking for many rare diseases and which prevents the development of medicines for many. So it's really a prerequisite for any research and development, and you can test and explore the role of prognostic factors and biomarkers. You can also measure the impact of the disease for the patients. They can be used to count the patients for epidemiology studies, pharmacoepidemiology, confirming effectiveness and reducing uncertainty. They can be used to compare the care of patients, the care that the patients living in different countries or in different hospitals are receiving, and their outcomes. You can see the evolution of survival in different countries, and that can generate hypotheses on why. Is it because some medicines are not used, or they are not used the same way, in a more prophylactic way in some countries, and to treat acute diseases in other countries, which can explain the differences we saw 20 years ago in the survival of cystic fibrosis in Scandinavia versus other countries. These are useful comparisons that can be made. So in terms of organization, in theory, the registries have no additional consultations, visits, or exams. It's part of routine medical care, and there is no need to travel to remote clinical trial sites. So they are convenient for patients who don't want to be engaged in more complex settings in clinical trials. One key question which really puzzles many patients is the need to reach 95% exhaustivity, or if you have too many patients rejecting the proposal to join a registry, then it could pose validity concerns. There are different purposes for these registries. They vary, and which data need to be collected or don't need to be collected. That's really where we need to involve patients earlier and more in these discussions. Also, the possibility for patients entering directly data in these registries, should it be first validated by healthcare professionals who are telling us that they don't want to be overwhelmed by a new flow of data coming from patients, from connected devices and wearables. So how do we do that? Can we enter data with maybe algorithm that would validate the data and not the investigators or the clinicians? That remains to be tested. Patients don't fully realize everything that can be achieved based on real-world data and registry data. So just two examples. here Flatiron that explored and measured the outcomes in Castleman disease treated with a monoclonal antibody or not, using a machine learning selected real-world cohort. They are not the only one who do this kind of analysis, but we as patient organizations need to understand more how these studies are done and how to interpret the results, the bias, etc. Otherwise, we have more difficulties to explain them compared to explaining the results of clinical trials, which are in a way more straightforward. Another example, target trial emulation for real-world data analysis to support HTA decisions. So this was research done in the HTX project, where the question was whether erythropoietin-sparing agent had an impact on survival in myelodysplastic syndrome, and depending on the protocol, and you could have an effect or not. So in fact, the effect here was positive, the survival was longer, only if the protocol, if the hypothesis for the data analysis of the registry, one assumption was that patients take the product, the ESA, with no interruption as long as necessary. In that case, the simulation, the emulation showed that there could be an effect. And that's the kind of analysis that we need to learn. They're not straightforward again, but with more training, we are confident that patient organizations can explain to their members this other analysis that can be made of our data. To finish, 2 words on some efforts. There are efforts in all member states, and there will be more. So this is just one example in France, where for rare diseases, there is now a national database with more than 1,600,000 people with a rare disease registered in this database. And patient organizations were involved from the beginning in all aspects of the development, including which minimum data set should be used to inform the system of the different diseases, the different patients. And there is now a module where you can add a minimum data set on treatments, not for all patients, not for all situations, but and minimum data on the name of the product, the dose, the route of administration, the duration of treatment, efficacy and safety, and for which purposes, in which situations will this minimum data set on treatments be used to measure the effectiveness of off-label use in rare diseases, to conduct some real-life pharmacoepidemiological studies, and also in the context of their therapeutic use protocols for early and compassionate access programs as a start, not to overwhelm the system, knowing that the clinicians who enter the data are paid according to the quantity of data they need to enter in the database. And with this, we have more than 95% of data completion in this database, which then can be extremely helpful in these registries to compare different treatments for off-label use, in particular when the use is so established, so generalized that you cannot even think of which randomization could be proposed to patients, that we need to rely on registry studies to explore these off-label uses. There are training activities, as I said, they are necessary. Patients should be consulted on which data should be in the registry. For example, in the multiple sclerosis registry, it would be excellent if, in addition to the relapses and number of relapses, we could have standardized data on the severity, the impact of the relapse. Which questions, again, involve patients to decide which questions are relevant for the registry and their studies. Scientific advice, we think it should be systematic as soon as there is a scientific advice to explore, to look at existing data sources, because later in the development or in the evaluation or after the authorization, you need to decide which registry studies would be necessary. And there is a lot of sometimes preparatory work to make sure that the data will be entered in these registries. The patients should be part of the governance of these registries. They should be in a position to approve the procedures to grant access to data, and they should take part in the decision-making of these registries. And finally, the feedback, whatever the complexity of analysis and the results, we patient organizations should explain to study participants, to patients, the results and their consequences. And there are cases with the simple infographics that can really help achieving this. So final considerations, the registries are also useful to define the new unmet needs, patients who do not respond to newly available treatments. So from the famous Get Real picture of all the possible uses of real-world data, I added one here, the updated unmet needs. We know that even with the best treatments, there are always patients who do not respond for cystic fibrosis. Not everyone can respond, depending on the genetic profile you have, to newly authorized medicines. So we need to revise constantly the unmet medical needs, and the registry data are useful to achieve that. The questions that are not yet fully answered: who should fund this research? Should we have multi-pharma registries like in CF or multiple sclerosis? Should we have a pay or play model? Either you pay for these registries to be in place, or you take part in these registries. The compassionate use programs, the early access programs, the off-label users are real-world sources of data too, and we think they are overlooked. And for registry studies, as for clinical trials, patients should be engaged in their design, in the conduct of the study, in the interpretation of results, and in the information to registry participants. And with this, I thank you for your attention.
Thank you very much, François, for this excellent presentation. And I think it shows that everybody has a key role to play in advancing real-world data and moving into from real-world data to real-world evidence. I think in the interest of time, we can take one question, and maybe the easiest one to take is from Devender Danda, which is who is asking what key rare diseases are covered in the French database, and how the database is available to use by a wider audience.
All rare diseases are represented in the registry. I think the objective is to have all people living with a rare disease registered in France, and they have already started to conduct very precise epidemiological studies on some rare diseases, but there is no limit in respect with which rare diseases can be registered. And regarding access, well, I think it's now for all data sources. The data are fully anonymized in the national data bank, so access is granted as per the European regulations to those who request access to the data with a data hub and data controller. All these procedures are in place in France, so I think the data are quite open. The data sources are quite open. They are managed by the Filière Manadira, which is a kind of network of all centers of expertise treating patients with rare diseases. They manage this database.
Thank you, François. And last but not least, I think we can move to the next speaker, with Kit Rose, with wearing multiple hats in academia and regulatory settings. And maybe you can tell us which hat you're wearing today. And you are here with Fayad Bergas to give the next presentation. Thank you, Kit.
Yeah, thanks. Thanks a lot, Cecile. I'm primarily wearing my academic hat, but it's very difficult to actually get rid of other hats as well. So, Bergas will share the presentation, and we will shift gears to some extent, in the sense that we will be looking at the use of registry data, not so much for the final decision making at the regulatory HTA phase of development, but rather early in development. And as I mentioned, I will introduce, and then Bergers will share with you the scientific work we've done in this area. Next slide, please. And picking up from what Carla already presented, the More-EUROPA project intends to cover the entire drug development life cycle. And up till now, we've been discussing very much the regulatory and patient perspectives. We will move towards the decision making early in development of new drugs, where also registry data can play a very relevant role in our view. And as it's important to share, we built much of the research and development within More-EUROPA on very strong case studies. So it's based on registry sources which are quite well developed. So it's a heart failure registry from SWEDHF at the Karolinska Institute, the big MS multiple sclerosis registries across Europe, and oncology related quality registries in combinations with hospital data. And I think what I particularly liked on François's presentation, if we ever need another incentive to build such good quality data, I think François has provided ample reason to do so. But we have the benefit here of profiting from registries that are already there. And if you typically use, next slide, please. If you typically look at where do we see registries used, and this is the set of potential uses from somewhat of a regulatory perspective, but also there throughout the drug development life cycle, it actually quite matches what also patients expect from registries in the sense it's understanding the disease, it's understanding the epidemiology of disease, but also evaluating impact of treatments once they are available for patients, either on or off label. What we've seen also, Kelly and Anna look at, is the use of real-world data in regulatory decision-making, augmenting the late-stage clinical trials or clinical evidence, and use them for primary decisions. As you can imagine, from decision point of view, those are the more challenging objectives. What we will be looking at is to what extent and how can they be used in early decision-making during drug development. Related to, for instance, when and in which subgroup or for which population to enter late-stage development. Also there, registry data can be of value, and we will illustrate that. Another thing that we will be looking at, but not discussed today, is at the same vein, once you have your trials and trial data in, registries can also be quite valuable in better predicting on how new treatments will behave in a broader population than was investigated during clinical development. So those are uses which are not primarily regulatory decision making, but where the availability and the data from registries can have true value. So then I will switch gears and leave it up to Bergers. We will be looking on how could we do that in guiding decision making and moving into phase 3 development with the SWEDHF registry as a real life example. So Bergers, the floor is yours.
Yeah, thank you, Kit. I'm just going to make sure that I am not muted. Yes. I will continue for the discussion on how registry data can be incorporated in drug development decision making. And I will talk about the probability of success. And I will give an example where we used SWEDHF registry data, and then I will show you a key learning and some takeaways on this study. So yeah, we are focusing on the decisions whether to move to phase 3 from phase 2 or not. And there is a quantitative measures to assess the uncertainty, which has been widely used. It's called probability of success. What is probability of success? It is a probability that the trial will show a significant treatment effect given a prior belief or prior informations on the possible treatment effect. So the prior distribution, the prior informations is in a form of prior distributions, which is called design prior. And if you are familiar with power, so POS is power, but average over a possible value of from the distributions, which many people also call POS as Bayesian power because it includes prior distributions. So in calculating POS, usually companies use estimate from phase 2 as the prior belief. It is straightforward if the endpoint used in phase 2 and phase 3 trials are the same. But we have a challenge if the phase 2 trial, let's say, use a continuous biomarker endpoint, while the phase 3 use a hard clinical survival endpoint. And then, yeah, how can we translate or use the information on biomarker from phase 2 to get information on survival endpoint in phase 3? Well, that's where registry data can be used or can be incorporated. So we have information on biomarker in phase 2, but we want information on survival endpoint for the phase 3, a planned phase 3. We can use registry data since in registry data, both endpoints are collected. And we use registry data to obtain the associations between these endpoints, between the biomarker and the survival endpoints. And it can be obtained using, for example, a Cox proportional hazard model, which we also used in our example. So I will show an example case where we use SweetHF registry data to obtain the association between NT-proBNP. So that's the biomarker used in phase 2. So we are using a case of a cosmic HF trial as if we are planning the phase 3, which is the galactic HF trial, and they use NT-proBNP change from baseline at week 20. And yeah, we will use the SweetHF registry data to obtain the associations. To give a context, cosmic HF trial was conducted in 2013 until 2015. It was a randomized double-blind study on the patients with heart failure and reduced ejection fractions. It's a 3-arm studies, and the drug that was tested is omecamtiv mecarbil. And yeah, the primary outcome was maximum concentration of omecamtiv mecarbil in plasma, and the secondary outcome, they measure the biomarker, which is the NT-proBNP, and exactly they measure the changes from baseline at week 20. This is the biomarker that we are going to use to translate to a survival endpoint. So one of the results is that it seems promising that in PK titration group versus the placebo, there's almost 1,000 reductions, 1,000 picograms per milliliter of NT-proBNP. Yeah, so that was the phase 2. Now we are going to talk about the one in registry data. In registry data, there are important decisions to be made. The first one is the timing of the follow-up and the biomarker measurements. And the second one is the patient populations. And then we need to consider or be careful on the data availability and missing data. And additionally, for survival endpoint, we can also choose type of endpoint. I will explain one by one. So the timing of the follow-up and biomarker measurements. So what I mean by timing of the follow-up is the period in registry data. Let's say here we have registry from January 2000 until January 2025, like today. Oh, now it's February, but maybe the registry is now until January 2025. And there is a phase 2 study conducted in 2023 until 2024. So we need to decide which period of registry data to be used. Should it be matched with phase 2 study, or should it also include a bit longer backward retrospectively? That's where we need to discuss with the expert on the disease, on the clinician, with the clinicians and the expert of the registry data. And yeah, besides that, we need to determine the follow-up for each patient as well. In phase 2, maybe each patient is follow-up for 2 years or 3 years, and in phase 3 it's 3 years or 4 years. And for registry data, should we use the same as phase 2 or phase 3 or other follow-up time? We need to discuss it with the expert on registry data and expert on the disease as well. And then also the biomarker measurements. In our example, we use the same biomarker measurements as the one in phase 2 study, the cosmic HF, which is the changes from baseline in NT-proBNP at week 20. And then for other settings, we use 3 years follow-up time, and we use the data from January 2000 until August 2015. That's the end of the cosmic HF, the phase 2 study. And now, the second important thing is the patient populations. Well, in registry data, we have broader population characteristics than in RCT. Yeah, it includes patients with extreme condition as well, like a more severe condition than the one in RCT. In general, match the population characteristics in registry to the characteristic in phase 2 or phase 3. But we also want a broader range of characteristics, because that means our estimation of association is more reliable if we have more broader characteristics. We could also consider possible subgroup of populations. I will give you an example. So in our case, we are going to look at the same patients, which is patient of heart failure with reduced ejection fractions, HFrEF. But we also consider other subgroups. So in registry data, we have patients that is generally treated versus patients that has all the necessary drug for patients with HF and reduced ejection fractions. During that time, there were 3 mandatory drugs for REF patients, but some patients only have 2 of them. So those are the general patients, and some patients have all the 3, and we refer to them as optimally treated patients. So we are comparing these 2 subgroups. And then the next one is data availability. In registry data are much more collected irregularly than in RCT. We have different follow-up times among patients, and this will affect other important aspects as well, like the timing of the follow-up and biomarker measurements. To account for the sparsely collected data, we apply some window of measurements. I will show you a visualizations later. We also need to deal with missing data. Should we do multiple imputations or other technique of missing data handling? That also needs to be discussed with the clinician. In our example, SWID-HF example, Cosmic HF used the baseline changes from baseline at week 20, but in registry data, all patients, well, not all patients, almost no patients have exactly record at baseline and record at week 20. So we apply a window of 10 week, plus and minus 10 week. Here is the visualization. In RCT, let's say patient 1, we have nicely collected data at week 0, and then at week 20, maybe a little bit of window here of 1 week, and patient 2, the same week 0 and week 20. But in registry data, let's say the second patient, this person has observation at week 0, but he doesn't have observation at week 20 exactly, but he does have observations at week 25. That's why we put a window of plus and minus 10 weeks for that to account the changes, measurement of changes from baseline. And then let's take a look at the first patients as well. The first registrations was at week 0, let's say, but here the NT-proBNP records are missing. The NT-proBNP wasn't recorded, so we don't have the true baseline value for NT-proBNP, and we need to put window of measurement as well. Or by some predictions or multiple imputations handling. That also needs to be discussed with the statistician and the clinician and the expert of registry data. And other example, the third patient, this person has observation at week 0, but the next observations is after 3 years, after the maximum follow-up time that we decided. So we don't have records around week 20. It needs to be discussed whether this patient should be excluded or we use missing data handling. And the fourth case, it's not of a problem as in the third case, because we have the survival outcome here. We just censored here, and then we also have the NT-proBNP changes. And the last one is the type of endpoint. Well, generally, we need to use the same endpoint planned for phase 3 study, because we want to see in phase 3 study if it works or not, if it's gonna show a significant effect or not. For survival outcome, we can take a look at different endpoints like death or hospitalization or composite outcome. And for CVHF example, the phase 3 study, the Galactic HF and the Galactic HF, they use a composite outcome of cardiovascular death and heart failure hospitalization. But we also try to look at other endpoint, which is the cardiovascular death only, and we compare these two, and we'll see what's the probability of the pause of these two outcomes, two endpoints, sorry. So as a recap, some important decisions include timing of the follow-up and biomarker measurements, determine the period of this the study in registry data, determine the follow-up time for each patient, and then also the timing of the biomarker measurements. For the patient populations, generally match the characteristics in phase 2 or phase 3, but we need to retain some of the broader characteristics in registry data and also consider other subgroup populations to explore. And then data availability, we need to apply some windows, and we need to discuss how large the window is with the clinician and the expert and apply it. And then type of endpoint for explorations. Generally, we use the same endpoint as the one in phase 3 study, but other possible endpoints could also be interesting to see. And this is the settings that we use for the SWEDHF data. 3-year follow-up, changes at week 20, and then this is the period, and then the patient populations, we take a look at HF patients with reduced ejection fractions, and then we compare the generally treated patients and versus the optimally treated patients, and we use the window of 10 weeks for the plus and minus 10 weeks for the changes of NT-proBNP at week 20, and we compare the composite outcome and cardiovascular death. And this is the estimate from Cosmic HF, the phase 2 study. And what is the pause? This is the result of the probability of success. So this is the patients and the duration, the period of the study. And initially, if we only look at the patients, all patients of HF-ref, reduced ejection fractions, in this period, we have 32,000 patients, roughly. But due to data availability and some missing data, we decided to exclude some of them, and we ended up with 1,000 patients, or even for optimally treated patients, only 380 patients. Even when we look at the effective sample size, which is the number of events, it's even lower, even 33 worth of informations in optimally treated patients with CV death outcome. And this is the resulting pause. Between the composite, there are not much changes, but between the general and optimally treated patients, the subgroups, it's almost tripled or doubled. the probability of success. So that's interesting to see in registry data. So yeah, some takeaway, discussion among clinicians, statisticians, and experts on registry data is very important to decide on the important things. We want to match the settings with the one in RCT without losing data. What I mean by that is use as much information as possible, as the data allow. And also we need to consider some different settings or scenarios for sensitivity analysis, whether the pause will change much or not. And thank you. I will give the floor back to Cecile, perhaps.
Yes, thank you very much, Kit and Bergas. I haven't seen any direct question yet to your presentation, but maybe before moving to the panel and the Q&A, there is one question, and I don't know if Kelly or Kit can take it, but it's a question from Thomas Klopstock, who is asking, can a registry receive EMA qualification before it starts enrolling patients, i.e. qualification on basis of the plan structure of the registry? Thank you.
Thank you. Kit, should I go ahead or?
Yeah, please do. I was kind of considering this to be the first question in the Q&A we are entering now.
Okay. Perfect.
Yeah.
So, yeah. So the for the EMA qualifications, there are two possible outputs. One is the opinion, so which is based on mature application. So this would be the case where the registry is, is well established, has collected a lot of data, already run some studies, and there is also possibility to request EMA qualification advice. So this would be, for example, it could cover this aspect of the registries that are still under development and not necessarily data collected. We had the case with the World Hemophilia Gene Therapy Registry, which came for advice. And actually, they got a letter at the end highlighting the areas where the registry should focus on in order to, continue the development, to improve it for future use for regulatory purposes. So it's possible. I hope this answers the question.
Thank you. And yeah, Kit, I think you have the floor now as a moderator. Thank you.
Yeah. Thanks, Cecile. So the floor is open for questions throughout, which you can pose in the Q&A box so that we can see and can address them. Maybe as I see your hand raised, Cecile, but I'm not sure whether that was intentional.
Yes. No, sorry, it's while people ask questions. I think that Kelly, during her presentation, highlighted a point that maybe is worth commenting on, and maybe also to have your perspective, Kit, on that. It's the context of use, which define the scope of a qualification. And I wonder if this is a concept that could be elaborated upon when we gather some questions. Thank you.
Yes, thanks, Cecile. It's actually a very crucial part of the qualification indeed. Kelly, you first, or shall I give a first reflection? You're on mute, Kelly.
Yeah, I'm just saying I was distracted, so sorry if you can repeat the question. Sorry.
Cecile raised the point that for qualification, the context of use is a very important concept to qualify against, if you like. So, and any thoughts there? Maybe also some experience there.
Yeah, and I think until now, so again, I was referring to this workshop we had last year on patient registry, and there was one day focusing on the qualification of this kind of data source. Maybe what has not been so clear until now is that really the qualification should be, the application for qualification is based on the context of use. So it's based on how the data will be used for regulatory purposes. And it can be, for example, for post-authorization studies, for external control arm, but the application should be based on the future use. So we do not qualify the data infrastructure itself, we qualify for context of use. And this will be made really clear in the updated guidance, which we will publish hopefully by Q2 2025.
Yeah, thanks. I think that clarifies and maybe what came up in earlier discussions. If you have a very narrow context of scope of use, it might be easier to qualify, but you might not be leveraging the registry to the full extent. So I think there would be an invitation there to think very carefully if you're developing a registry to make most use of it. Probably you don't want a too small context of use. That's the kind of for registry developers. I'm also giving a few seconds for the floor to come up with examples. Maybe Francois, you've covered quite a bit of ground, but I think one of the things, at least that resonated with me, is the value of information. So you shared, I think, an interesting experience where indeed a fairly small data set was sufficient to market the drug conditional marketing authorization. Maybe to extend a bit on that thinking, we've also seen examples where that was done even without the conditional, but under exceptional circumstances. So your reflections and what you see, so it could be conditional, but then you still have an obligation to get more data, but even that might be challenging in some settings. So you might be aware of the ALS example where actually one of the considerations was, well, that additional information might actually be impossible to gather.
Not sure I'm familiar with this specific case, but the condition can only work if you can generate additional evidence. So in other words, the access to the product should be granted, otherwise you will never. And I think this is where we need to have this discussion on the value of information, not just with regulators, but HTA and payers as well. They need to understand that in an ideal world, we would like the highest scientific evidence that we would like to get, but the reality check is that you need to decide and you need to, it's a trade-off. Do you have enough information today for a decision to be made? If not, what would be the cost? And not only the financial cost, but all the other costs, including the time to defer the entry on the market of a potentially effective treatment to obtain this highest quality evidence.
Yes, thanks. Yeah, I see a hand raised. Sita, please.
Yeah, thank you, Kit. There is still a question in the Q&A, which might be nice to discuss here in the group. It says there are questions from EMA and HTAs around the concurrence of real-world cohorts for ECA purposes. Can you shed some light on how this should be evaluated and addressed, as if there is possibility of concurrence in ECA? There can also be RCT. And the person who asked this question would like to hear your views on this.
Yeah, I'm seeing who to give the floor to address this question. I would be tempted to start again with Kelly or Anna, but
I actually wrote some pieces of response in the chat.
okay.
I'm not sure I fully understood the question, but I was saying that if there is a plan to use external control arm, I guess this is what ECA mean, right? This needs to be properly planned and designed in order to make sure that the ECA is as comparable as possible to the interventional arm in terms, for example, on the patient characteristics, the baseline data, the timing of collection, the endpoints, making sure that these endpoints are properly collected in the real world data source. And again, for this, there needs to be a prior assessment to make sure this is properly done and can be used. For this, applicants can come for scientific advice in order to, discuss with regulators on the possibilities. So, yeah, I don't know if that, covers or addresses the question. And I
possibly see that in the response if I could add one perspective, which I think would be helpful for all of us. And that's partly on terminology, but also partly on what's good practice. So in my thinking, I would probably speak of an external control arm, if it's actually planned and prospective.
Yeah.
Because that's for me is the control part. So that it would actually be in the original protocol of the trial design. And then you can actually solve quite a few of the key questions that Kelly raises prospectively, which makes the evidence much stronger. The setting where you have a post hoc data augmenting your trial is a fundamentally different one. And I think we would be helped to make that distinction, and if at all possible, start moving towards prospectively planning at the trial design stage. I think that will be very helpful, and also make let's say the acceptability as confirmatory evidence much more likely.
Kit, may I ask to confirm as well, is there a plan for guidance on external control arm planned at the level of the MWP?
Yes, the quick answer is yes. We are just composing the temporary drafting group to do so. So that's something that will happen start happening in the course of this year, and most likely we will have a specific workshop organized around it.
Great.
To discuss some of these, I think, very important ideas.
Perfect.
Seems we've answered the question, which is great. Thanks, Kelly. Yeah.
Thank you.
Any more from the floor? I see Cecile's hand raised.
Sorry, it's on this topic. I wonder if there is the opportunity to clarify the use of external control as part of a single product development application at the time of licensing, and the qualification of a registry as external control. What are the differences and expectation from the regulators?
Should I start or?
Yeah, I think this kind of seemed logical. Yeah,
Good.
It's also not a surprise that somewhat from the regulatory you get in the middle of the of the discussion. Yeah, please go ahead.
So qualification of a registry to be used as external control arm, I think there was one application, which is the Huntington registry, if I'm not mistaken, and I think you will find that in the slides, in the last slide of our presentation. So obviously, in order to come for qualification for this use, there would be the need to, present the data of a proof of concept study, etc., just to show, the characteristic of the registry data to be used as external control arm to a specific interventional arm, so to a specific product. So so that would be, an evaluation which is done based on the data submitted in the qualification. But if there is an application for a new product in this same indication, there will still need to be an evaluation, like another evaluation, like a feasibility assessment based on a specific research question. So even if there is a qualification for a patient registry to be used as external control arm, there will still need to be an evaluation in the context of the new application to be sure that the external control arm is still relevant, fit for purpose to be used as such in the evaluation of the new product. I don't know if that's
Thank you.
That's responsive.
Yeah. Thanks. I see Francois' hand raised. Please, Francois, go ahead.
It's a short question on the external control arms, but some organizations have requested access to the data from the placebo arms of completed studies as a source of external data. Is this something that is in the scope of the guidance on external control arm?
Real good point, and the quick answer is yes. And also, if you look at the FDA existing guidance on external controls, I think there's also reference there, and it's even seen as a somewhat preferred source of comparative data, because you're still the data is also originated from a clinical trial setting. So there are advantages are seen there, and I think the sharing of data, I think, is the underlying thing that we really need to push for. I will just quickly pick up a question that we get from the floor. Should the rationale for selecting a registry over another be provided as part of the feasibility study? So should the feasibility study actually be somewhat more open? That's how I read the question, to the data source to be used. Opinions are welcome, but it's kind of also logical to once again to look at the regulatory folks here.
I think that's an interesting question, to be honest. I mean, before starting a study, maybe I can also go back to a very important aspect of, regulators always prefer the use of patient registries over what we should not call a product registry. So meaning that we would prefer, using data from existing registries, which collect data not only on a specific product, but on all the products indicated in the indication, or for the disease, or even non-treated patients. So I think what is always a good practice to do before starting a study is really doing this kind of research on what are the existing registries out there which can potentially be used for the study, and to assess each of them how they can best address the study question. So I think in a way, in a feasibility assessment, this is what is being done. We try to identify what is the best registry to collect the data and to be used for a specific study. So I think it will always be interesting for regulators to see, benchmarking of what is the best registry. I don't know if that is used in practice, if a feasibility assessment is mostly used based on one registry, or if there is also data or information provided about the other registries, but that would be useful, I think, for me personally, just to make sure the best registries or either one or several registries are used.
Yeah, thanks. Or even combined, or as a test case to have some replication. So it would even be interesting to see that the answer replicates across different registries. That would even add additional value. So I think it's an interesting question and concept to think about. Cecile.
Yeah, maybe to add also, to link with the More-EUROPA project, our last webinar was showcasing a tool that is developing, leveraging AI on how to find more easily registries in order to inform the decision making in drug development or post-marketing setting. And maybe I would encourage to look at the previous webinar of More-EUROPA. Thank you.
Yeah, that's a good reminder, Cecile. And Justin, and we will move to wrapping up soon. But in a previous project, IMI Protect, very much focused on safety. There we had the privilege of being able to answer the same safety-related question across different registries, and that proved to be actually quite valuable in terms of solidness of the evidence we were generating. Given the time, we have 3 minutes left, at least according to my clock, but I think we share the same clock worldwide nowadays. I will try to briefly wrap up. First of all, thanks for all the speakers and the presentations. I think what we've roughly seen, starting with Kelly and Anna, is first of all, that we do see an increase, however modest, but in the place that real-world data and real-world evidence is taking in regulatory decision-making. My take on it is we're in a learning phase, and what we develop in terms of guidances, but also the interactions we more actively seek on the concrete cases, I think they help us learning more efficiently and more focused. So I think we're getting there. I would almost say, as per usual, I think that Francois has highlighted, I think from a patient perspective, but actually more general, how crucially important it is to actually make sure that we get the data to be able to do the research, and certainly in the interest of rare disease patients, because there is a huge gap there. And I think we didn't touch upon it here, but also the subtle, but I think real situations where we actually have to rely on real world evidence, because really RCTs are simply not possible in some of the settings, but the considerations are actually quite subtle. And a clear call to do our utmost of gathering high quality data for research purposes. And finally, Berg has expanded on a somewhat different, but I think valuable use in actually estimating success early on in development and leveraging registry data there. So I think we've kind of seen an interesting case, largely, at least from the More-EUROPA perspective. It was actually ongoing research to some extent, which also sketches the way we are seeking interaction. So this was happy to have this webinar and then engage, and as Cecile mentioned, we will back with more webinars. So thanks everybody for your attention and your contribution.
Thank you very much.
Thank you.
Thanks. Bye.